We're going to move on and talk about a topic now that at first can give students a lot of trouble, but it's a really, really important concept and one you need to understand because it describes the events that determine how things move from the blood out into the tissues and how other molecules move from the tissues and back into the blood. This is the process of capillary exchange. Now, capillary exchange is really a blanket term for a number of different processes that are all happening at the same time in a capillary bed. And the four main ones that your book talks about are diffusion, transcytosis, filtration, and reabsorption. So let me define those for you real quick. Diffusion, you will remember, is the movement of molecules from an area where they are in high concentration to an area where they are in low concentration. And generally speaking, the rate of this process is slower than for two more other processes I'm going to talk about in a minute called filtration and reabsorption. Diffusion can happen across a capillary through a couple of different routes. One is, if this is my cross-section, my capillary, remember there are those intercellular clefts, those little spaces between adjacent endothelial cells. We can have diffusion of things from where they are in high concentration to low concentration or vice versa, high here, low here, through those intercellular clefts. And we can have things diffuse directly across the plasma membrane if, for example, they happen to be lipid soluble. So, for example, oxygen is a lipid soluble molecule when it's by itself. If it's just an oxygen paired with another oxygen, it's lipid soluble. It can pass right through the plasma membrane, through the cytosol of the cell, and out the plasma membrane on the other side. That would be diffusion. Transcytosis is another process by which we can move things into and out of capillaries, and that involves the formation of a vesicle. And that vesicle then carries the item across the endothelial cell and then exocytoses it on the other side. So this is transcytosis involves endocytosis, transport across the cell, and then exocytosis on the other side. Now, these next two terms, filtration and reabsorption, are really two different variants of a concept called bulk flow. And bulk flow is basically anytime I can, through a passive process, move a large number, a whole bunch of molecules in the same direction together, and I can do it at a speed much more quickly than I could do with diffusion. And usually this is because I've got some sort of fluid pressure, and in this case, for example, it could be the pressure of the blood itself is literally forcing forcing molecules out of the capillary and into the surrounding tissue via bulk flow. If that bulk flow causes things to move, items to move from the blood out into the tissues, it's called filtration. If that bulk flow instead causes things from the tissues to move back into the blood, we call it reabsorption. The filtration process we're going to see on the artery end, near the artery side of a capillary bed. And this is how we deliver things like oxygen, amino acids, vitamins, nutrients, etc. to the tissues. On the venule end of the capillary bed is where reabsorption would occur. And this is where we get the tissues to deliver to the blood carbon dioxide and other metabolic waste products that we want to clear from the tissues in that area. So the process of capillary exchange is obviously, as I've already mentioned, specialized for the movement of things like oxygen, carbon dioxide, and different types of nutrients. And the movement of those, again, as I said, principally happens by diffusion, but it can also happen by bulk flow, and it can also happen by transcytosis. Now, just to remind you, with diffusion, if I'm lipid soluble, then I can just diffuse down my concentration gradient via movement right through the cell, right? I can go right through the plasma membrane, through the cytosol, and out the other side. Non-lipid soluble items, for example, also or have to rely more on these intercellular clefts usually. Maybe they have to rely on fenestrations if we're looking at fenestrated capillaries. Sometimes they rely on protein channels, and sometimes they also use transcytosis. Now, one key thing to remember is that large proteins like albumin, globulins, fibrin, and all the red blood cells still remain in the blood. They do not move out. It's only small things, oxygen, carbon dioxide, amino acids, glucose molecules, nutrients, like vitamins and minerals that will have a tendency to move in and out of the blood via capillary exchange. Larger items do not do that. So let's take a look at the actual physiologic dynamics that contribute to the movement of items into and out of the blood. The key here, the number one most important thing to remember here is the role that blood pressure plays in this. Remember I told you that there is a pressure gradient between the aorta and the superior and inferior vena cava. And that pressure is highest in the aorta just as we're exiting the heart. Pressure is lowest when we're returning to the heart at the superior and inferior vena cava. That means if I'm looking at a capillary bed, here's my capillary bed, and this is the arterial side, here's the venule side, that pressure by definition is going to be slightly higher on the arterial side, slightly lower on the venule side. That's going to be important in a minute. When I show you the next slide, you're going to see how that plays out. But the number one reason why we get the filtration or the movement outward of items from the blood and into the surrounding tissues on the arterial side is due to the fact that the blood pressure is slightly higher there than it is at other locations along the blood vessel or the capillary bed. Now, blood pressure isn't the only thing that we're going to see on the next slide that comes into play with determining which way fluids move into or out of the capillary. There is another pressure, another fluid pressure that also contributes to the behavior of capillary exchange, and that is something called oncotic pressure. Some books call it colloidal osmotic or oncotic pressure. Now, if you talk to a research physiologist, they would probably go to excruciating lengths to distinguish very carefully between osmotic and oncotic. There are very slight nuances of differences between those two terms, but for the purposes of our class, we're going to think of them all as the same. Colloidal osmotic, oncotic pressure, just think of them as the same for purposes of our discussion. And what this is the amount of pressure or draw a particular solution has to water. So if you remember, a solution is made up of a solvent, that's some sort of fluid such as water, plus solutes. Those are particles that are found in that solvent, that are dissolved in that solvent. So if we look at the blood, it has all kinds of particles in it, amino acids, proteins, and then it has big old red blood cells all over in it. All of these different particles present in the blood are, and it's mostly the proteins that do it, they're the biggest contributor, contribute to the colloidal osmotic pressure of the blood. And the more particles I have present, the greater I will have an ability to draw water into the blood. The fewer protein particles I have present in the blood, the less I will draw water into the blood. Okay, so this is colloidal osmotic pressure. Blood that's very highly concentrated, like if somebody's dehydrated, it's going to have a very high colloidal osmotic pressure. That means it's going to draw water pretty vigorously. Okay, so the dynamics of capillary exchange that I'm going to take you through on the next slide is principally going to be the result of kind of a tug of war between the blood pressure and the colloidal osmotic pressure. Okay, so keep that in mind, blood pressure and colloidal osmotic pressure. And one other thing I should tell you before we go to the next slide, relatively speaking, colloidal osmotic pressure remains constant. Pretty much always more or less the same. However, we remember here blood pressure as we move through the systemic circuit, it drops. The farther we get away from the heart, right, the lower the blood pressure is. Farther we get away from the left ventricle, I should say, the lower the pressure gets. So even though there's this interplay here, this tug of war going on between blood pressure and colloidal osmotic pressure that I'm going to show you, ultimately it ends up being blood pressure that really determines the behavior of capillary exchange, because it's the only one of the two that actually changes over the length of a capillary bed. Colloidal osmotic pressure really doesn't. So here's how this looks. And we will review through this in class too, because it's a concept that's tough for students to wrap their heads around at first. So just let me orient you first. Here we're looking at a very simplified rendition of a capillary bed. They just represented it by a single blood vessel, but you can imagine that there's a whole bunch of capillaries in this capillary bed. Here's the arterial end of the capillary bed. Over here on the right is the venule end of the capillary bed. So when blood exits an arterial, goes into a meta-arterial, and enters into the capillary bed, we can, at that point in time, at this location here, we can measure just how strong the blood pressure is and just how strong the colloidal osmotic pressure is. And if we take these two pressures and we add them together, it should give us some overall indicator of which way fluid is going to move. Is it going to move out of the blood and into the tissues, or is it going to move from the tissues into the blood? So let's see how this plays out. We're going to look at blood pressure first, which is the first part of this table right here. And collectively, your book calls this hydrostatic pressures, which means pressures of the water itself, okay, the water that's found in the blood, etc. Now, if we look at the hydrostatic pressures, there are two different pressures coming into play here. One is the actual pressure of the blood, and that's due to, the ventricle contracting and the aorta expanding and recoiling, all that good stuff. All of that contributes to blood pressure or blood hydrostatic pressure. On the arterial end, we can measure that as being about 30 millimeters of mercury pushing outward. Okay. Now, remember that out here in the tissue, there's something called extracellular fluid. That's that fluid that surrounds all cells in our body. So it goes. It should make sense to you that because there's pressure out there, fluid out there, that fluid is generating a little bit of pressure that's going to be pushing inward towards the blood. And there is a slight amount of pressure pushing inward. It's actually not pushing inward, technically speaking. It's actually pushing almost like a vacuum. Okay, and here's why. We haven't learned about these things yet, but there are these special types of vessels. Notice I didn't say they were blood vessels. I said they're a type of vessel called a lymphatic vessel or lymphatic capillary. These are part of your immune system. And along with their duty of transporting white blood cells around in the body and a few other things that they do that we're going to learn about shortly, one thing that they do is they suck up, if you will, for lack of a better term, they sort of vacuum up extra fluid that begins to accumulate out in the tissues at these capillary beds. And so extra fluid gets sucked into this capillary bed, which is a good thing, because if it didn't, you get fluid accumulation in your tissues and you get edema as a result. So fluid kind of gets vacuumed up by these lymphatic vessels, and eventually that fluid will be returned back into your blood at the level of your subclavian veins, up underneath your clavicles. So this pressure of the fluid being drawn into the lymphatics actually creates almost a kind of like a little bit of a suction effect, with a value of about 3 millimeters of mercury pulling fluid into that lymphatic. So here we go. This is called your interstitial hydrostatic pressure. That's a result of those lymphatics kind of having that suction, a very, very, very weak suction effect on the fluid. So as a result, we have blood pressure pushing out with a value of 30. We also have the lymphatics sucking or vacuuming the fluid out of the extracellular fluid with a value of about 3. So if I add those two together, the overall push is to have hydrostatic pressure or fluid pressure pushing outward out of the capillary on the arterial bed with a value of roughly 33 millimeters of mercury. Okay, now that's half the story. Remember I said on the previous slide that there were two main players here, the pressure of the fluids like the blood pressure, and then the other big main player was colloidal osmotic pressure, the pressure of generated by having all these particles present in a fluid such as blood plasma. So now we need to look at that colloidal osmotic pressure and see what contribution it's making to this process. So again, we're still on the arterial end of the bed here. And if we look at colloidal osmotic pressure, we can see that it's a composite of two things. The blood has particles dissolved in it, so there's going to be what's called a blood colloidal osmotic pressure. And that's going to have this draw, this pull on water to pull it in to try to dilute out all those proteins and red blood cells, etc., present in the blood. It tends to have a pulling in effect of pulling water into the blood with a value of about 28 millimeters of mercury. Now, out here in the extracellular fluid, we also have a few proteins and other types of solutes that are just dissolved out in the extracellular fluids. So the extracellular fluid itself is also going to pull water in its direction to try to dilute out its concentration of little particles present out here. And it tends to draw water towards it, which would be out of the blood and into the tissues with a value of 8. So it pulls with a value of 8 out. So we've got colloidal osmotic pressure wanting to pull water into the blood with a value of 28. We've got osmotic pressure in the extracellular fluid trying to pull water out of the blood with a value of 8. Okay, so 28 pushing into the blood, 8 pushing out. Add those two together, the overall colloidal or net colloidal osmotic pressure is to try to pull water in with a value of 20 right here. So if we consider those two values, the hydrostatic pressure value and the colloidal osmotic pressure value, and we look at those, we've got one of them wanting to push fluid out with a value of 33, push fluid out of the blood, and then we've also got another one wanting to pull water in with a value of 20. If I add those two together, what I find out is there's a net movement of water out of the arterial end of the capillary bed with a value of roughly 13 millimeters of mercury. Okay, now let's go over to the venule end of the capillary bed. Now on the venule end of the capillary bed, we still have the interplay of these two pressures. We still have hydrostatic pressures coming into play, and we still have colloidal pressures coming into play. Now, the colloidal osmotic pressure here actually does not change because I still have the same number of particles present in the blood as I had on the arterial end, and I still have the same relative number of proteins, etc., that are in the extracellular fluid out in the tissue. So I still have the blood wanting to pull water in with a value of 28, and I still have the tissues wanting to pull water out of the blood with a value of 8. So I still have an overall push or drive inward of fluid into the blood with a value of 20, just like I had before. That didn't change at all. What did change is that because I've gotten a little farther away from the left ventricle, my pressure has dropped a little bit. So whereas on the arterial end, my blood pressure was pushing fluid out with a value of 30, now look, on the venous end, it's only pushing fluid out with a pressure of 10. We still have the lymphatic system serving as sort of a vacuum effect to pull water out of the blood. So the net result is I have a total drive out of the blood from hydrostatic pressure, a value of 13 pushing out. But I have a value of 20 for colloidal pressure pulling in to the blood. If I add 13 out plus 20 in, I get an overall drive of fluid into the blood, into the venous end of the capillary bed with a value of about 7 millimeters of mercury. So key things to remember here. Colloidal osmotic pressure remains the same along the entire length. The thing that changes, the thing that determines that items get forced or filtered out on the arterial end and reabsorbed on the venule end is because of changes in the blood pressure. Right there. That's the key. Blood pressure drops. Therefore, we go from forcing things out of the blood on the arterial end to pulling things into the blood on the venule end. Makes good sense. On the arterial end, we can push out things like oxygen and different nutrients, amino acids, glucose molecules. And then on the venule end, we can pick up carbon dioxide, metabolic waste products like urea, uric acid, creatinine, other breakdown products from metabolic activities.